Okay. Hello everyone, good evening and welcome to the NPTEL course. I'll just share my presentation. Okay. So this is week 2 of the NPTEL course, which is BT60, which is Introduction to Proteomics. So what have we done in week 1? I think a few of you were there in the last lecture, like in the week 1 lectures. What did we do in our first lecture? If anybody wants to recall it once. Yes, Aishwarya and Arup, I think you were there in week 1 lecture 2. What did we discuss? Yeah. So basically, in week 1, as Aishwarya said, so we gave an introduction to amino acid, introduction to proteins, how proteins are getting folded, the rules and regulations and stuff, how we can denature it, how we can renature it, then a bit of introduction to proteomics, a brief overview of it, and we did a question and answer session as well. So in week 2, it is largely based on gel-based proteomics. So in that gel-based proteomics, we'll be learning how exactly. So before learning the technique itself, like any type of proteomics, no matter what type of proteomics you are doing, uh, you need to collect the sample as accurately as possible and also prepare the sample for the analysis depending upon the type of instrument you are dealing with or depending upon the type of analysis you want to do to identify your protein of interest or protein as a whole, whatever your aim might be, depending upon your aim in order to study a protein and depending upon the technique you will be utilizing to study that protein, your sample preparation will vary. So in this week 2 lectures, most of the part is related to sample preparation, how exactly what all types of precautions you need to take, what all type of contaminants you need to remove, how can we remove that, how essential it is to have a purified protein when you are doing the analysis. All those will be handled in week 2 lectures. So we'll be dealing with sample preparation and few pre-analytical factors and as well as extraction of proteins and after extraction, how can we quantify them? And we'll be dealing with one specific two techniques basically, one-dimensional electrophoresis and two-dimensional electrophoresis. So initial part is about how we make the sample. Then after, if we are making the sample, how can we analyze? By using gel-based proteomics. In that gel-based proteomics, one thing is one-dimensional electrophoresis, wherein you will be using SGS page itself. And we'll be introducing two-dimensional electrophoresis. We'll not be completing it though, just introducing what 2D is. So how do we separate proteins in the first dimension and the second dimension? How it enhances the analysis of a protein that we'll be dealing with in part 5. Okay, so that is the whole thing in week 2. So the people who were there in my last lecture, they know that. So what I do is I generally give an overview of whole 5 lectures which were uploaded in week 2 now. So I'll be just giving an overview of everything, all those 5 videos which you might have gone through. Then we'll move ahead with certain question and answer session. Okay? Yes, people, can show thumbs up. Okay, so let's start. So before moving on to proteomics or gel-based, in general, proteomics can be studied in various techniques basically. No matter, so basically, studying proteins is nothing but proteomics, right? But that specific proteomic analysis can be done by various techniques Today we are just dealing with gel-based method like SDS page and two-dimensional electrophoresis. There is DIGE as well. There is native page too. And for that analysis to visualize them, we need certain stains like massive blue stain, silver stain, or fluorescently labeled stain, or pro-Q diamond, etc. These are different staining and different. All stains have its own property to visualize certain type of protein or certain concentration of protein. So that will change. Even there are some gel-free based mass spec as well. So these are few gel-free based techniques. We'll not be going in detail of that. So we in this course basically you are dealing with gel-based as well as mass spectrometry. You are not dealing with gel-free mass spec or interactomics or structural proteomics. So we'll be dealing with this first section and the third section. And in terms of mass spec, the protein has some is fragmented with the help of ionization source. And after fragmentation, those masses are analyzed by using different mass analyzers. That's how analysis takes place in terms of mass spec. And it is if you are doing MS like fragmentation after first fragmentation, then it becomes much more accurate analysis of those proteins. Then interactomics is about studying if a protein is interacting with a DNA or a lipid or a protein itself. Those type of studies can be done with the help of interactomics. And if you want to study a structure of a protein, X-ray crystallography is one of the technique which is generally used to understand the structure of a protein. For that, you basically require a purified protein. You need to crystallize the protein and then subject it to X-rays, and there will be a pattern generated, and that pattern is analyzed using certain softwares. That's how a structure is predicted. Similarly, there is NMR, electron spray, crystallography, etc. So all these techniques are responsible for like help in understanding structure of a protein. So as I just mentioned, like we will be dealing with gel-based method and mass spectrometry only. So coming to week 2 lectures, so that's I just wanted to give how proteomics can be studied. Before doing everything, we need to have sample. We need to know the source of the sample. How can we collect those specific protein sample, and how can we extract proteins out of that specific sample. So there are different methods for it. So we'll be dealing with the sample preparation. So first thing is in the first lectures I discussed about generally a protein will be produced inside a cell, right? And we need to lyse the cell and take out the protein outside first. So first thing would be obviously lysing the cell. And generally inside the cell, protein is generally protected. Like there are protease inhibitors present, etc. But as soon as it comes out, it might be unstable, and we need to make sure that whatever proteases which generally degrade the protein are present, we need to make sure that those proteases are inhibited by adding certain protease inhibitors or something like that, so that the protein which is coming out is not getting degraded. So protection from proteolysis is also essential, and in that as soon as you lyse it, it has everything. Like it has DNA, it has protein, it has RNA. You need to make sure that all those are separated, no contaminants are present, and you just want the protein to be analyzed, right? And in that protein also there will be all sort of proteins present. Like inside a cell there will be a number of proteins being produced. So it will be having each and every type of protein, but you might like depending upon your aim. If you are studying the whole, then fractionation might not be required. But if you are studying certain type of protein or only one specific type of protein, a certain class of protein, then accordingly fractionation would be required by using different techniques, and that would help like enhance your understanding about studying that specific protein. So the sample source can be anything. It can be the entire organism, just a cell, fluid of it, like from an organism or tissue from that organism. So make sure that specific collection of source is highly reproducible so that whenever you are doing any analysis, like you can reproduce that depending upon your like proteomic application require different approaches for sample preparation, as I mentioned earlier. So depending upon your aim, if you want to study the protein as a whole, in everything about whatever might be present in your sample, that's called as global proteomic analysis. If you are trying to study only, for example, you have engineered that specific organism and engineered for expressing certain type of protein. And if you are trying to understand whether that specific protein is overexpressed or not overexpressed, that become expression proteomic analysis. Along with that, for example, in one specific sample, you want to see few types of protein, whether those are uh, present in a disease condition and not present in a normal condition. Those type of analysis called as target. Like I want to check about this specific protein only. That's targeted proteomic analysis. So that is done only like targeted proteomic analysis also deals with quantifying it. So whether and global will not deal with quantifying it because you are studying each and every type of protein. You will not have standards for each type of protein and you will not be able to quantify it. You can have a relative analysis like this protein is more than that specific protein. That can be done with global proteomics, but you cannot exactly quantify it. But in terms of targeted ones, you know what type of protein you want to analyze. If you have standard for that, then you can quantify that specific protein as well. That's targeted proteomic analysis. So whenever you're doing sample preparation, that's the procedure involves solubilization, denaturation, reduction. Basically, you're treating the sample in this, and this additional step will improve your protein extract. And sometimes adding these steps may need to lead to loss of selective proteins as well. You know, to make sure that you're doing the steps at one go and avoiding any external things to interfere so that your protein is not getting degraded, neither it is getting lost. All those precautions need to be taken care while preparing samples for protein analysis. You should ensure that whenever you are extracting, whole of the protein is coming out and not part of it. And there will be interfering compounds. So make sure you choose a method which will allow minimal interfering compounds to be present. And once it is there, you need to ensure that you need to remove them as well by adding. So we'll be dealing with that. How can we remove that? And good protein extract can ensure that. So if you have a purified one or like minimalized interfering compounds, then your further proteomic experiment would be successful, much more successful comparatively. So this is the protocol generally. You need to solubilize it. You need to make sure you prevent from aggregation. You need to denature and reduce all proteins so that you can analyze it, remove all the nucleic acid contamination, and remove other contaminants like lipids, carbohydrates, etc., or salts, which might be present in the buffer. All those needs to be removed. So, so you need to remove nucleic acid salt. You need to make sure that whenever you are preparing the sample, just after preparing the sample, if you are not doing the analysis right away, you need to store the samples at -20 in aliquots. And once that is stored, make sure that whenever you are ready with the analysis technique or method, etc., then only remove it for thawing and then do the analysis. Repetitive freezing thawing can degrade your sample. So make sure you don't do that. So good sample preparation was like all the proteins should come out. It should be possible that you need to like reproducibility should be possible. So make sure that that the source where you are collecting the protein is available and you can reproduce the results. And initially you need to optimize the protocol with lesser number of samples. Once it is started working, then you can go ahead with large number of samples. So protein extraction from source material, solubilization, and make sure that all those interfering components are removed. So this is the workflow. You need to lyse the cell, protect it, fractionate it, extract the protein out of it, remove the contamination, and quantify it. So why do you need cell disruption? Obviously, it's present inside the cell, and you need to take out those proteins from the cell. So that's the reason you need cell lysis. So to facilitate effective disruption of the cells, as well as to isolate proteins from intact cells and to obtain all the proteins in the sample, and also helping in minimizing the sample recovery and making sure that the protein structure is maintained. So that is only possible by choosing an appropriate buffer which has all the components essential to make sure that protein is stable after coming out. So there are different ways of lysing. These are certain gentle lysis method or else chemical based method like adding some hypotonic solution so that because of the osmotic pressure it will burst out, adding certain detergent or adding certain enzymes which will degrade the membrane and everything will come out. Just do freeze-thaw cycles again and again, and because of the external pressure changing in the temperature, the cells will lyse and the all the components will come out. That's gentle lysis. There are mechanical or physical based lysis as well, such as sonication, manual grinding, French pressure, homogenization, etc. These are certain harsh method, and this is required for some samples which have cell wall or whose cell membrane is much more thicker, and it is very difficult to lyse just by gentle lysis. Then you need to use these methods for completely lysing the cell. Then it will release proteases as well, which may result to. So you need to make sure that you need to add protease inhibitors so that those proteases will not degrade the protein of interest. So these are few examples: PMSF, EDT, AGT, etc. So this is responsible for serine 16 proteases. It will inhibit that, and these are responsible for metalloproteases. So that's lysis. So you need to make sure that all those components are there in your while doing the lysis, either gentle way or harsh way, depending upon the type of source you are having. Then fractionate is possible to isolate the protein because if you are not doing global proteomic analysis, fractionation is required. Even if you are doing global, fractionate and analyze one by one and then compile it together because analyzing whole thing together becomes a little difficult and the machine is also not that uh, efficient enough when you have a lot of protein in like basically a complex protein mixture in one uh, specific sample. If you have just isolated by doing some technique, like we'll deal with fractionation technique, and it would then if you want to do global proteomics, you can just compile all the results and represent your data accordingly. But fractionation initial step would be beneficial for better proteomic analysis, and it allows improved resolution basically when an individual fraction is analyzed and it becomes less crowded 2D maps. So there are fractionation technique like centrifugation, sequential extraction, electrophoresis, affinity chromatography. So this will basically uh, fractionate type of, for example, centrifugation will mitochondrial proteins will be different, plasma membrane proteins will come in supernatant, etc. So that can happen. In terms of affinity, if you have certain affinity attached with the protein of interest, while doing chromatographic technique, that will be isolated and analyzing that protein will be giving you much better results. Electrophoresis separating on the basis of molecular weight or by pH, etc. So depending upon various fractionation technique, isolating your proteins would be beneficial for final results. So that basically increases the proteome coverage. And for example, if in a specific sample there are 2 3 proteins which are very highly abundant, and all other 100 proteins are there, but they are in minimal concentration. So generally, what is preferred is you need to remove that highly abundant proteins so that low abundant proteins are uh, visualized or detectable at least. If these three are also present in the same sample, then low abundant proteins won't be able to be detected as efficiently if you are not removing them. So removing them would be beneficial, and so that lower abundant proteins are also in a detectable range. So that's what was explained in week part one of the week one lecture. Then so we just did the how do we do lysis and how do we protect the protein, and as well as fractionation. Then now we have fraction sample. You need to extract protein out of it. So you can do various technique by subcellular fractionation. You can so it's there, but it's along with the buffer while doing the lysis. It's not just the protein; it has some other contaminants also. So you need to make sure that all of the contaminants are removed and the protein is as concentrated as possible. So concentration can happen with the help of precipitation. There are different ways of precipitating and removing the salts, removing the DNA, RNA, etc. So proteins naturally form complex with membranes and nucleic acids. So they are generally there together. So you need to make sure that only protein is present in a sample and all other are removed. So that is done with the help of solubilization by using certain components like urea or thiourea. Thiourea is the one which improves solubilization of membrane proteins. So if there are some proteins, if you want to study membrane related proteins, make sure you add thiourea to your fractionated sample so that that membrane proteins get uh, solubilized. Similarly, urea is added. It helps in stabilization, unfolding of proteins, and giving ionizable groups to the protein. Basically, overall reason is solubilizing the protein. You can also use some detergents, which is extremely effective for hydrophobic proteins. So if your sample has more amount of hydrophobic proteins, use SDS and make sure that your further analysis is not isoelectric focusing. If it is there, then don't use SDS because it's not compatible with IEF. So that's what the thing is like, depending upon your whole picture, the type of sample, the type of analysis you want to do, then the sample preparation will change accordingly. Mhm. No, it won't. So IEF technique you can directly, so that you can just directly add your lysate itself, and it has a mixture of proteins in terms of IEF. So when we are dealing with IEF, maybe I can explain that how exactly what type of samples are can be added and how it can be used. Okay. So so SDS is basically not compatible with IEF. So you just asked whether urea and thiourea is compatible. So I'm not exactly sure whether they are compatible or not, but according to me, they might not be. So, because SDS basically gives negative charge to the protein, and urea, I don't think even that would be possible because it will give certain charge to it, and that might change the isoelectric point of the protein. So, that is the reason I feel that maybe IEF not, for doing IEF, you cannot add urea as well. But I need to confirm whether, yeah, but I need to confirm whether that is compatible or not. But I feel it is not compatible. And similarly, some other detergents like similar to that of SDS. So this is butyl ionic detergent. It prevents non-specific aggregation, and basically it helps in all those are these different components depending upon the type of protein you are dealing with and depending upon the type of analysis you'll do. You can use different solubilizing agents to solubilize your protein. Then there are certain reductants as well. So this reducing agents cleaves disulfide bonds like DTT and TBP, and these are non-ionic reducing agent, and this is the most common reductant which is used, and it basically breaks the disulfide bonds. And you can use ampholites as well, which is also present in IP like IPG buffer, and it is added during sample solution prior to IEF. So what do? They basically minimize protein aggregation and also enhance protein stability before doing the analysis like isoelectric focusing. So you can add ampholites to it, and this is basically minimizing proteolysis and help solubilization of the protein. So these are the few uh, what components you can add during the sample preparation itself, depending upon your analysis. So you need to ensure that protein solubility during extraction and separation. So typical sample solution example is this thing having DDT, some detergent as well as urea, and so that a complete solubilization of protein will take place. Whereas depending upon the type of sample, like plant cell will require harsh method, even fungal cell will require harsh method for lysis, whereas body fluids might require gentle lysine and abundant proteins need to be removed. Body fluids, like all this might require uh, gentle lysis, whereas these might require harsh lysis. And each and every type of cell has a different composition. Like this might have a lot of interfering substances. So you need to make sure that the contaminants are removed by doing repetitive steps of the same thing so that all the contaminants are removed. This is having proteolysis problem. This is having nucleic acid to protein ratio is much more greater. So nucleic acid needs to be removed completely. So each and every source of sample might have different issue with it to get a purified protein. Accordingly, that specific step needs to be much more stringent so that those components are removed. Then some pre-analytical factors like prior to that, so prior aims simultaneous analysis of thousands of proteins. So what is an impact of pre-analytical factors which you need to consider because prior to the point of actual sample analysis, this is very high for proteome scale clinical studies. So what do we mean by that is in terms of pre-analytical factors is for example you have a disease sample, you have a normal sample and you are trying to compare. For example, you are having both normal sample, but in one of the sample you found that few of the proteins were highly abundant, whereas in one of the sample few of those proteins were less abundant. You just collected the sample, but you don't know any pre-analytical factor. Then you won't be able to analyze. You just know that those two individuals were normal. We just took their blood samples, did protein analysis, but still you found some variation. If you don't have any pre-analytical factor along with that, then you won't be able to do any analysis. So pre-analytical factors are essential, such as some intrinsic factors like gender, age, ethnicity, diet, medication, smoking, alcohol consumption, etc. So all those pre-analytical factors you need to make sure that you have all of those stuff so that after getting the analysis and after knowing the results, you can connect that specific thing. So, for example, if a person is alcoholic, that specific protein might be highly expressed compared to the one who is not alcoholic. So that change might you might see in terms of doing certain analysis or a disease state, a normal state. That disease state will have certain type of protein in high amount, whereas normal might not have that. So you need to have that pre-analytical factors in mind so that your analysis becomes much more easier. So factors such as patient posture, application time, at what time you're taking it, whether you're dealing with serum or plasma. So there will be difference in serum and plasma also. And how you are collecting it, how you are storing it, how you are handling it, all might have some effect in your protein analysis. Like this is about storage, avoid multiple freeze thawing, avoid long-term storage. You make sure that once the sample is collected, you are doing the analysis as fast as possible. So there are different pre-analytical factors. You need to make sure that you have all of those stuff and then do the analysis. Then, in order to concentrate the protein, there are different methods. One of the method is ammonium sulfate precipitation. So, this due to high salt what happens in this technique is basically due to high salt concentration. This is salting out basically. Protein loses water in their hydration cell because externally there is high amount of salt and that makes aggregation of the protein and it precipitates out because of high salt concentration present outside. And so, depending upon the type of protein, depending upon the type of protein, the amount of salt as you increase, different proteins get precipitated out, and that's how you fractionate it using this technique. And at one specific salt concentration, you need to centrifuge it, then it get precipitated out, and take the supernatant, add more amount of salt, and again repeat this process until you get your protein of interest. That's how fractionation takes place in terms of ammonium sulfate precipitation. Similarly, there is acetone precipitation as well. So this can be like you need excess of at least 3 or 4 volumes of ice cold acetone. So this process is done at colder temperature, and you need to incubate as so that's basically a normal procedure, and it helps many organic solvent contaminants like detergent lipids to remove by using this technique. Similarly, there is trichloroacetic acid precipitation as well. And in this method, 100% sample recovery is expected. So depending upon your need, you can accordingly use such type of techniques. You can include both of them as well, like TCA plus acetone, and it allow precipitation for like proteins are allowed to precipitate for 1 hour in this technique. And so basically, these are different ways of precipitating and concentrating your protein. You can use like there are some methods which are specifically most useful for some type of protein. So depending upon the literature, you can accordingly choose that type of method for your protein of interest. There can be ammonium acetate in methanol method as well. So as just said, like these are different methods to precipitate your protein or concentrate your protein and try and eliminate those interfering compounds. So this is used for plant samples generally. It helps in removing polyphenols. So as we were just dealing with the same thing again and again, depending upon the type of sample, type of analysis, your whole sample preparation technique changes. So this is specifically for plant samples. Then you need to remove all the contaminants like salts, small ionic compounds, polysaccharides, nucleic acid, lipids, etc. And if your aim is to perform two-dimensional electrophoresis, salt is most likely the reason for bad IEF. So if you are doing isoelectric focusing as your next analysis technique, make sure that you completely remove the salt. And that can be done with the help of dialysis or gel filtration or precipitation techniques, which will help in removing the salt. Generally, these sources have high amount of salts like urine and plasma. So you need to make sure those are removed by these techniques. If you are doing isoelectric focusing, if you are not doing that, then you can go ahead with the some other technique as well. Then dialysis is the most popular method for removing it. And in terms of removing nucleic acids, and it is essential to remove nucleic acid because if you are not removing it if while doing isoelectric focusing, it will give you streaking in the isoelectric uh, like IP strip, and because of the presence of nucleic acid, you need to make sure that those are removed, and those can be removed with the help of DNases and RNAs. We just need to add those into your sample so that DNA gets degraded, and if there is a presence of RNA, then that gets degraded. And in terms of if there is any presence of polysaccharides, so this is possible depending upon the type of sample you are dealing with. Then those can be removed as well using these methods: ammonium sulfate precipitation or phenol ammonium acetate precipitation or TCA precipitation. This will help in removing the polysaccharides. If there is a presence of lipids in the sample, generally if low amount of lipid is present, that's fine. But in the presence of detergents, like the solubilization, which we are the first step which we dealt with in terms of sample preparation, that should disintegrate lipids and solubilize protein. So these disintegrated lipids will automatically getting removed from the sample. So that if it is not still, delipidization is a method which can be used for removal of lipids. Like generally these are present in high amount in plant seeds, algae. If you're dealing with these samples for your proteomic analysis, make sure you do delipidization as well so that all the lipids are removed out of your sample. So these are ionic detergents. So ionic indicating giving certain charge to the protein. So these techniques cannot be used for isoelectric focusing. And these are some other interfering compounds, and accordingly there are methods available to remove them as well. So this is the whole summary of the same of week part 2 of the week 2 lectures. Then sir discuss one of the method whole method for bacterial sample. a bacterial proteomic analysis. So now we have done with how samples are prepared, how it is extracted, and how can you remove all the uh, contaminants from the sample of interest. Then in week part three of the lecture, what sir has taught is he tried to give one of the method which is Trizol method, which is used for uh, bacterial proteome analysis. So in that method, you are just using Trizol as an agent to uh, extract proteins of from bacterial sample. So what do you use? You can use for bacterial sample preparation as heat treatment. You can use precipitation method or direct extraction or use the with solubilization buffer that can be used, or you can use Trizol method as well. So what happens in Trizol method? It's basically you are extracting protein, and when you are using this method, it is able to recover DNA, RNA, and protein. So it happens in different sections basically. Upper layer has RNA, middle layer has protein, etc. So you make by using this technique, when Trizol is generally used for RNA extraction as well, but here it is as you are dealing with protein, you are trying to use Trizol for protein extraction. So in this method, generally you will never find nucleic acid contamination, or you will not require desalting because salt contamination won't be there because Trizol removes all of it. Basically, what Trizol has is it has guanidinium isothiocyanate, which is an inhibitor of RNase, and it also helps in giving you good quality RNA. But here we are dealing with proteins though, and it will generally, by using this method, lipid contamination is also not there because we use chloroform in this method, and chloroform is something which dissolves lipids. So generally, all those are removed by using this technique, and proteins are easy to resolubilize if these are not present. So what do you do in this method? You just add 1 ml of triazole, you add 200 microliter of chloroform, shake it vigorously and vortex it, and then centrifuge it. Then you will get a pellet. So you need to carefully remove the upper layer which has RNA. The bottom layer, to the bottom layer you need to add 300 microliter of ethanol, which will help in precipitation of DNA. Then that gets precipitated, and now your protein is in the supernatant fraction containing protein to collect into a fresh tube. Then the supernatant, the upper layer, you just need to collect it into a fresh tube. Add 4 volumes of chilled acetone. So as we just know that acetone is used for precipitation of protein, for concentration of protein. Now by adding this, the protein gets precipitated. Now you need to discard the supernatant and retain the pellet, and you can just wash that specific pellet because if there are certain salts or phenol contamination, etc., if it is present coming from trizol, those are removed, and just dry the pellet and reconstitute in the lysis buffer. Now you have your protein. Then you can quantitate that specific protein by UV spectrophotometer by by analyzing at 280 nanometers, or you can also do different assays like Bradford assay or fall in love assay, etc., to determine the protein concentration present in your sample. So I think we did do this in our last lecture, how the quantification happens. So that's what Trizol method was in part three of the lecture. So, so I just gave one of an example, like one of the method, which is Trizol method, but except that there are different types of fractionation method, different types of precipitation method. So depending upon the need, you need to accordingly select those and prepare your samples. So now your sample is prepared. In terms of gel-based proteomics, one of the technique is electrophoresis. For proteins, you can use either native page or SGS page. Even now, that becomes depending upon if you are having a complex protein and you need to know whether those complex is being formed or not, then you can use native page. Then you cannot degrade it. And if you want to, so after degradation, it will have certain fragments, right? Those fragments can be analyzed with the help of SGS page. Or else, if your protein is only linear, has single fragment and it's not a complex protein, Then you can directly do SDS page and try and check whether your protein of interest is present or not. So electrophoresis technique was invented by these scientists in 1930, and they developed this technique. And here the separation is solely on the basis of charge to mass ratio and molecular weight of protein. And they migrate as smaller one, are the one which migrate further and larger migrate slowly. And in this technique, there is native and SDS page. Both have their own benefits. So electrophoresis for protein characterization, that is SDS page or native page. So it's a single dimension technique wherein large number of proteins can be separated with good resolution. Cell lysate can directly be loaded, so you don't need to have those fractionation, and even if you don't have that, still you can do the analysis by using SDS page, precipitation method, etc. So what does SDS page do? It's a widely used electrophoresis. It gives negative charge to the protein, and you can also use some uh, reducing agents like beta mercaptoethanol so that it denatures the uh, disulfide bond, and it will be much more easier to separate the proteins on SDS page. What happens in this? How how is this specific gel made? By using bisacrylamide and acrylamide. So this is the cross-linking agent, and that's acrylamide is the main matrix, and this will cross-link only in the presence of APS and TIMID because this initiates polymerization, and this gives the as a stabilizer effect, and it promotes polymerization. So in the presence of these two, this cross-linking will take place with acrylamide, and then they will solidify. Then while sample preparation, you add SDS to your sample. You can add beta mercaptoethanol to your sample so that protein gets a negative charge and also breaks the disulfide bonds. And once you do that sample preparation, you need to heat it so that SDS and beta mercaptoethanol works. It gets negative charge, and then the separation can take place. So lower molecular weight separates faster compared to that of higher molecular weights. And you can either use ladder or you can use certain known molecular weights like these standard proteins that accordingly you can compare and check whether your protein of interest is present or not. This is how silver chained protein looks like, gel looks like, and comassive chained gel looks like this. So basically, SDS binds at a ratio of 1.4 gram of SDS per gram of protein. So accordingly, SDS needs to be used, and it gives uniform charge to mass ratio. So that is the reason, like after the addition of SDS, it gives uniform charge to mass ratio of all proteins. So because of that, the separation is solely on the basis of molecular weight and not because of charge. Whereas in terms of native page, even charge is involved, but still it gives a negative charge because of comassive dye. Whereas SDS gets negative, sorry, SDS page gets protein gets negative charge because of SDS, whereas in terms of native page, protein gets negative charge. So in this, basically, sample is mixed with comassive blue dye to provide necessary charge to protein complexes, and then they are separated. Unlike SDS, so dye does not denature the protein here. The comassive blue dye will not denature the protein, but it binds to them in their native state itself. So it is not denaturation happening here. So if there is a complex protein that can be identified by this technique, so that's the application basically using native page. So it is determined by negative charge because of comassive dye. And now the separation is on the basis of molecular weight, though, but still the size and shape of the complex will also influence the separation. And that's what was talked about in part 4 of week 2 lectures. I hope you all are getting it. Yes, I think so. In the part 5, so talked about just give an introduction of like two-dimensional gel electrophoresis. So as the name suggests, it's two dimensions. That is first dimension and second dimension. So you have your protein sample. You need to first do isoelectric focusing, which is called the first dimension, and then before taking that first dimension into second dimension, you have certain steps to uh, do it. That is equilibration of IPG strips. So once you do equilibration, it is getting ready for SDS second dimension analysis. Once you do that, then you do staining, you do image analysis, spot picking whether your protein of interest is present or not present. If it is there, how much it is greater than the control. All those analysis can be done. And if you find something relevant to your type of analysis, you can just cut that gel, then cut the protein fraction of that present on the gel, take it, do enzymatic digestion, and can give it for mass spectrometry analysis by so that will have certain molecular weight. It will get ionized. Then, depending upon mass to charge ratio, m by z ratio, it will get separated. The mass analyzer will try and analyze the mass, and you can determine whether your protein of interest was actually there or not. It will further confirm basically using mass spec analysis. So what are the objectives of doing 2D gel electrophoresis? Study global and differential protein expression, resolve proteins from complex mixtures, study isoforms and post-translation modification if there are any, and visually analyze the protein. So what happens in this? So if I give you a broad overview of the analysis, what happens in this? So you might be knowing isoelectric point. It is a point at which protein has net zero charge. So at that zero charge, it stop moving. Until and unless it gets the zero charge, it will continue to move on this IPG strip. And as soon as it reaches a pH at which it has zero charge, it stops. That's how these proteins are. So this everything has a complex protein. Now we'll after doing the first dimension, you equilibrate the sample, denaturate the proteins, etc., present in the location itself, and then those can be separated again using a second dimension, which is SDS page, and you can accurately analyze what type of proteins were present and in the at that specific PI. And so basically, doing isoelectric focusing and uh, then doing SDS page helps you to separate or fractionate protein much more efficiently. Or else, if you take the same sample, do SDS page, all of these proteins will be present in one lane itself. So initial separation basically separates them depending upon the PI, and then separating in the second dimension helps you to fractionate them in a much more better way by doing 2D gel electrophoresis. If you do just one dimension directly SDS page, all of these proteins which you are seeing in a separated way in different lanes, you might see that in one lane itself. So that is the benefit of doing 2D gel electrophoresis. So protein separations according to isoelectric point, that's the principle of isoelectric focusing. And so it is basically proteins introduced into immobilized pH gradient. So the strip itself has pH gradient, and to separate on the basis of isoelectric point. It is the point at which it does not move because it is OV, because it doesn't have any charge. So what's isoelectric point? It's basically the point at which it doesn't have any charge. Equivalent to 1, and if it can have either negative charge or positive charge. So it can be either having the positive charge or a negative charge. If they have plus and minus both, then it has a neutral charge. So it's nothing but a measure of tendency of a group to give up a proton. So either giving a proton here or giving a proton here, and with the tendency decreasing, 10 folds as pK increases by 1 unit. So here it is increasing by 1 unit here, and that's how. How can you identify uh, PI of any specific amino acid? So this is the amino acid titration curve. By uh, adding bases to it, the protein charge will keep on changing it. and that's how you can determine isoelectric point of a specific amino acid. Then each amino acid has its own charge. Then the whole protein might have different amino acids with it. So you basically take an average of all of it in order to find a specific pi of a protein. So this is how the separation happens. At low pH, it is generally positively charged. It will go try and move towards the negative one. At high pH, it generally has negative charge, and it will try and move towards the positive. Uh, try and move towards the positive electrode, and that's how the separation will take place. And it is there will be a point at which it will have both. So this is just an example. For example, the red protein here, it was positively charged, and it went on to move till it gets that one specific negative charge, and it stopped while. So it is uh, getting its deuterionic form where no longer it has two charges, but no longer it has one charge, either positive or negative, and it stops. Similarly, the blue one stopped here. That's how it moved. Initially, it had a positive charge, and then uh, the protein stopped. Whereas this protein initially had negative charge, it went on to move till it gets the positive charge and becomes neutral, and it stopped moving here. So whenever it stops, it's basically nothing but a net zero charge is taken place for that protein, and it stops moving ahead because there's no charge present. So at lower pH, proteins are generally positively charged. At higher pH, uh, proteins are negatively charged, and then they will accordingly move until and unless they find the isoelectric point. So that's your isoelectric focusing. Then you need to rehydrate. So this protein extracted from previous step, initial what we have just done in terms of preparing your sample. After that, you need to rehydrate the IPG strips overnight in a reswelling tray. So there was a lab session video. If you have gone through those lectures, you can imagine those trays and how exactly this protocol is taken, like practically done. So if not, you can go through those video lectures where uh, one of the lab member has shown those technique, how it is exactly performed in the lab. Then you do passive rehydration where no voltage is applied. We do active rehydration where a low voltage is applied, and you overlay a mineral oil so that the strip gets rehydrated first, and then this is just the overall review. Then IPG strips basically have different pH ranges. So depending upon the type of protein you want to analyze, different types of IPG strips are available, 4 to 7, 3 to 10. Depending upon your need, you can use that type of IPG strip. Different lengths are also available. If you are having highly concentrated protein, a lot of protein, use the larger size so that it gets separated much more efficiently. If not, you can use smaller size IPG strips as well. So it changes depending upon your need, and accordingly, you will need a larger uh, SDS page also if you are using 24 centimeter IPG strip. And larger ones generally become much more tedious. So you need to balance it basically. So 7 centimeter generally require this much amount of protein to be loaded. If it is greater in size, more amount of protein needs to be loaded. Maximum volume is also written here. Similarly, stain which you will be using for staining it, uh, that will be accordingly this much concentrated to analyze the protein. So that's the first dimension, separation in isoelectric focusing on the basis of isoelectric point, sample loaded on the strips after overnight rehydration. So first you do rehydration and then load your sample and then run it, and they will migrate on the basis of their isoelectric point. So after first dimension, as we just discussed, we need to equilibrate it. So condition step where proteins are separated by IF. So now we need to code the protein with SDS for molecular weight basis separation. So now it will give a negative charge to all the protein. So initially there was no charge, right? Because they stopped when you did isoelectric focusing. So now those specific proteins will be treated with SDS so that they have negative charge now, and then they will be separated on the basis of molecular weight. So what does this SDS do? It clears the intra-chain disulfide bonds. You also add DT, like reducing agents. So So SDS and reducing agents both. This is the mixture you add, and so you do equilibration twice for 10 minutes, so that complete equilibration takes place. Then the second equilibration is with the help of alkylating agents and reducing agents. What do they exactly do is so this is the first reaction in terms of second equilibration. First is SDS, which gives negative charge. Then alkylating and reducing agent, what do they do? So this, when you add the reducing agents, it breaks the disulfide bond. So if you see, initially a protein was dipolypeptide chain. By removing this disulfide bonds, it got broken. Here it got denatured. So you need to make sure that these disulfide bonds are not getting joined back. So in order to avoid that, you add acetylated iodoacetamide so that it gets acetylated, and there is no free sulfide group in order to make disulfide group again. So that's the reason alkylation is step is also added so that they do not get renatured and form the disulfide bond again. So that's what is being discussed. So this was just an introduction till the of two-dimensional electrophoresis till the step of equilibration. So we just did the first dimension. What's the protocol? Just a minute. So we just did till equilibration. From week three lectures, we'll be dealing with the second dimension. How do we do staining, image analysis, etc. So we just did first dimension, which is isoelectric focusing, and what equilibration is done and why it is done basically. Then these steps we will be dealing with in the next week lectures. So that's the overview of whole five lectures which sir taught you all. Our videos are being uploaded for that. So before going ahead, like before dealing with question and answers, maybe we can take a break of 5-10 minutes or so, and then we can start with the question and answers. Till then, if you are having any doubts with whatever I taught you all, you can ask me, or else we can take a break of 5-10 minutes or so. Yes, Prabha, you want to ask something? You have unmuted yourself. Like, do you want to ask anything? Okay, so mute yourself. Thank you. Are you guys back? Yes, should we start with question answers? How do I get the screen that I can see the chat box? Just a minute. Yeah. Okay. You can see the screen, right? I just yes. You can attempt this question. So there's a standard curve which is being made using bovine serum albumin by using Bradford method. So these are the samples PQRS, which shows an observance of zero point 0.4, zero point 1, zero point 0.3, and zero point 0.5. And this is the standard which was made, and the OD was taken twice, and this is the average which was found. Now we need to find the concentration of the sample. If this is the OD for Q, which is 0.123, what would be the concentration of the sample? Yes. Oh, okay. So there's an equation here. This is x-axis, y-axis, right? So you have a value of OD, right, with us, which is the y-axis. So you have this value 0.123 of Q, PQRS, right? So this is PQRS. That's what the OD is. So for Q, if the OD is 0.123, so Y is 0.123. Accordingly, solve this, you will get the X, and you can get the value of the amount of DSA, the concentration of the sample. If that is complicated though, like a bit, so you can just say that if 2 mu G is equivalent to 0.164, what would be 0.123? Just cross multiply and get the answer. Like it will give you approximate answer, but even that is fine for an MCQ based question. If 2 microgram is equal to 0.164 OD, what would be 0.123? Lesser than 2, obviously, because 0.164 is 2. 0.123 will be lesser than 2. So there is only one option which has lesser than 2 value. Yeah. So the answer is A. Similarly, so you need to find which following statement about the protein yield in decreasing order is accurate. Decreasing order indicating the highest concentration will come first, then the lower, and the lower. Decreasing order of protein yield. So OD is directly proportional to the concentration. So we have OD here. We don't have to find the concentration here. We just need to give the order. They have not asked us to find the concentration of each and every protein, each and every unknown sample, and then tell us. Obviously, OD is directly proportional to the concentration. You can just directly check the OD and you can tell the order. So this is zero point 0.5 first, which is S, then P, then R, and Q. So it is SPRQ as per the OD, zero point 0.5, zero point 0.4, zero point 0.3, and zero point 0.1. So in this, D option is the one which is giving you a correct order of decreasing order. So answer is D. Fine. Okay. So now triazole method, we discussed this. In a tube of extraction, which of the following subsequent layer she should take forward in order to extract RNA? So the question is about RNA, not about protein. After the first step, this is after the first step itself. As soon as you add triazole, you centrifuge it, vortex it, centrifuge it. How is the separation generally? The upper layer has what, middle layer has what, and the C layer has what? Yes, upper is RNA and middle is protein. Upper is RNA, yes. So which layer she should take for extracting RNA? It's obviously layer A. So this middle and in the C section that is DNA and protein, you then add ethanol so that DNA gets precipitated out. Then you take the upper solution which has protein and then you add acetone which will precipitate protein and all other contaminants can be removed out. So that's how the separation takes place when you are dealing with protein. Here the question is about RNA. So the upper layer you just take it which will have RNA and then you further process that for extracting RNA. So initial sample, so these are the options. Preparation is crucial stage for every proteomic experiment. For the best sample preparation, what should be ensured? It is prevented from repetitive uh, freezing and thawing. Nucleic acid are not degraded. Most of the proteins are solubilized and extracted. The presence of salt is maximized. Which one you should make sure that it's there? Yeah, the answer is C. So why are nucleic acid typically removed from samples before being in a gel-based proteomics study? Why do you think they are removed? 2, 3, and 4? Because nucleic acid being smeared through electrostatic interactions, the nucleic acid can bind with proteins and ampholetes. Nucleic acid with higher molecular weight may clog gel pores. Yes. Okay. In terms of SDS, gel element controls the resolution and separation of protein according to the sizes. Which SDS gel? Stacking gel, SDS concentration, acrylamide concentration, or running voltage. Controls the resolution and separation according to their sizes. It's C and not D. Running voltage will just separate the protein at a faster speed or a slower speed. It is nothing to do with the size of the protein or whatever. The resolution will change depending upon the concentration of acrylamide you are using. Some people use 10% gel, some use 12%, some use 15%. So making those gels, you add acrylamide accordingly. For making it highly concentrated, you add more amount of acrylamide. For making it less concentrated, 10% or something, you add lesser amount of acrylamide. So how does that help? So depending upon the acrylamide concentration, the pore size will change. So at higher concentration, higher molecular weight, for example, you are using 15% acrylamide, the higher concentrated molecular weight will take much more time to get separated, whereas the smaller molecular weight will get resolved much more efficiently. Whereas in terms of you're using lesser concentrated uh, gels, even the higher molecular weight uh, proteins will get resolved much more efficiently. Even smaller will obviously get resolved, but even higher ones will get resolved much more efficiently. So, depending upon the resolution, like depending upon the acrylamide concentration, the resolution and separation will change. You need to make sure that you use same concentration if you want to reproduce. So, C is the answer. I hope you are getting it. If not, you can let me know. Yes. So, which statement of one-dimensional and two-dimensional is true? You can just read the question. For casting a gel, timid is not necessary, although it is for one-dimensional. Yes. You said only two, or you wanted to say only three. So unlike SDS page, so it's not two and three, it's just three. So stacking gel is not necessary because in the first step itself, they are already aligned at one specific space. Like if you are having isoelectric like IPD strip, right? They are already stacked at one specific location and they can directly enter into resolving. Whereas when you are doing SDS page, stacking is required because the proteins are not aligned at a similar lines at the first stage, right? Then when they try to get stacked at one specific location and then they start resolving. So stacking gel is required for one dimension, but for two dimensional, the stacking is already done with the help of IPG strip, so it is not required. So three is the only answer, and two which separates protein. So it is not separates protein on the basis of mass to density; it's mass to charge only. So that's a wrong sentence. Sorry. Okay. 2DE, two steps are rehydration and equilibration. Which statement about this step is correct. So just before second dimension, they are saturated with SDS, and there is then second equilibration as well, which as alkylated and reducing agents. So this is for 9 and 10 both. So which of the following describes protein sample A that has not been reduced? So let's read the question. So that is A and B, two dimeric proteins. So it is a dimeric protein. So it was were isolated by Ruby. A protein molecular weight is greater than B. She used a reducing agent to modify the protein sample before running them. So the disulfide bonds are broken now. Okay, and A has higher molecular weight than B. Reducing agent to modify the sample before running them in lanes 1 to 5. So she has run the lane in 1 to 5 of SJS page to compare protein profiles of reduced and unreduced samples. So there are few samples which are reduced, there are few which are unreduced. So in one sample, so she is reducing agent before running the samples. So in that also it says that to compare reduced and unreduced sample. The representation of the gel image she acquired after gel chaining gel is provided below with different bands designated A to E. So these are different bands which are designated A to E. Which of the following describes protein sample A that has not been reduced? So disulfide bonds are not broken now. So which band do you think is representing protein A, which has higher molecular weight than B and it has not been reduced? That's what they say. So which band is representing that specific protein sample A? Yes, because that's the highest one, right? We cannot exactly say. So as per this question, it should be obviously A, as it is as they say that it's the highest molecular weight and it is not being reduced. So highest it should come at the topmost one. If it is reduced, then the same sample like this is 100. So one is around 70, one is around 30, right? So this third sample is nothing but the reduced protein A sample, right? Am I right? So yeah, so as per this question, the one whichever which has higher molecular weight that automatically becomes protein sample A here. Okay. So look at the previous. Which of the statement regarding protein A and B is accurate? So both A and B are homodimers. Both proteins are heterodimers. Protein B is homodimer while protein A is heterodimer. And protein A is homodimer while protein B is heterodimer. What do you think? So homo means same molecular weight or two fragments of same molecular weight. Hetero means two fragments of different molecular weight. So we know it for A. You can also get to know for B as well. You say C. So B is homodimer. That's the reason we just see one band initially. So this is not reduced one. This is reduced one, which is showing 25. This is showing 50. And accordingly, that's what helps you to get this answer. So those were the few questions for week two. I have few more questions as well. If we want, we can discuss that. Do you all want to discuss few more questions? Yes. Sorry. I had few more questions, but I'll check that and share it. The recorded ones, I think you have been provided this sheet like this. You might be having access to this specific sheet. So I add the YouTube links here. You can just go to that link and watch the recorded lecture. And this is the folder where you can find the presentations. I cannot find that. Just a minute, I'll try and find that and reshare it. How do I stop sharing? Okay, so this although these are from a different course, but it has similar questions which you can solve. A few of the questions are solvable, which is relevant to the lecture which we did today. Okay, so this is about native page and SDS page. So try and determine the answer. Read the question. So in the presence of SDS, it shows these bands. In the presence of reducing agents, it shows these bands. So what is the subunit composition of the protein? So reducing agent, after adding reducing agent, the band size changed to 90, whereas 160 and 60 are there when it is not being seen. So this DTT is doing something to this band, what it is doing, and what is the subunit composition accordingly. Yes. So okay, we didn't do this, so I'll just skip this. But still, if you want quantitative proteomics experiment, the preferred choice for large number of samples would be, so you need to quantitate as well, quantitative proteomic analysis, not just qualitative. Yes. So the question itself tells about that quantitative. So it's label-free quantification. Okay. So try and find which method you need to consider. So in a serum, there are high abundant proteins like albumin, globulin. You are working on a serum sample. You need to identify low abundant proteins. So which of the following method you will use to increase the coverage of those low abundant proteins? Which method will you use? A or C? So protein precipitation is there, yeah, but first step would be depletion of high abundant protein. This is the first step. And then you can go with the A method or ammonium sulfate precipitation, desalting, whatever. The first step would be obviously removing the high abundant one, and then with lower one for making it more concentrated, removing contaminants, etc. You can do all of these other methods. The first step is depletion of abundant protein. So you can do different method. Like for example, you are doing uh, what can we do in terms of removing albumin and globulin, high abundant proteins. I think so. You can do gel filtration, like gel-based chromatography, wherein higher molecular weight proteins will get eluted later, but all smaller molecular weight albumin or globulin are higher molecular ones. So that can be one thing. Or centrifugation. I don't know whether centrifugation will help. Uh, how can we do it? Like, so basically, your target will be depleting them. How can we do it? Is one of can be gel filtration chromatography. I think so because albumin and globulin are higher molecular weight proteins also, so they will get eluted later. As all other smaller molecular weight will get eluted first, and you can just mix them and do your analysis, and higher will get separated. What can be the other methods to do it? Maybe. mm, mm. No, 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 no. This precipitation happens like in terms of ammonium sulfate precipitation. It happens on the basis of the type of protein it is. So if you know albumin will get precipitated at what concentration of salt, you can use that method as well. But overall, what is your aim in terms of doing it? Depleting the high abundant protein, right? So that's the reason the answer for this question will be depletion of high abundant. That's what your aim is. What type of method you are using, that depends. But ultimate aim is to deplete the high abundant proteins. Right? Are you getting? Yes. So you want to study mitochondrial enzyme which is involved in beta oxidation, which are the method you will use for fractionation of subcellular organelles. Not sequential extraction. Sequential extraction will deal with protein, no matter at what organelle that specific protein is. But you are dealing with cellular organelles, you want mitochondrial enzyme only. So at different centrifugation speed, a specific type of organelle will get precipitated. So depending by changing the speed accordingly, you need to go through the literature like what speed mitochondria is getting precipitated, etc. And accordingly you can do. So you are doing subcellular organelles fractionation. So those organelles are getting fractionated depending upon the speed of centrifugation you are using. So centrifugation is the answer. So we haven't done system biology in this lecture, so I can skip that. Protein precipitation can be performed using which of the following solvent or all of the above? It's all of the above. The cross-linking agent in polyelectramide gel is acrylamide, timide, APS, bifacrylamide. Yes. So there are five proteins. These are five proteins. The A, B, C, D E have different molecular weight, different pi. So give the order of their migration from top to bottom of the gel. From top to bottom, higher molecular weight will run slower. At the bottom, there will be lower molecular weight ones. SGS pitch. So what would be the migration from top to bottom? EBCAD. Yes. Membrane proteins are highly hydrophobic in nature and thus difficult to extract. In this slide, which of the following reagent will be used for solubilization of membrane proteins? Which of the following stain? We didn't do about staining, we just did the equilibration step. So I might skip this question. So in blue native page, which is the like it gets its charge because of Lipids often interfere in proteomics. Which of the following organic solvent is used to remove lipids? Okay, it's methanol. Which of the following statement is true for SDS page? S. All of the above. Salts often interfere. How can they be removed? Yeah, that's fine. How are salts removed? Equilibration, gel permeation, precipitation. Should salts often interfere? Removed or which of the following technique is not employed? Is not employed for removing salts. So maybe you might have skipped that not here, hence you are answering D. Which one is not employed? Yeah. Which of the following pre-fractionation, aim of pre-fractionation? Yes. Increasing protein coverage, reducing sample complexity, removal of high abundant proteins. Before doing the analysis, what all is essential to do with the sample? It's all of the above. You need to do like remove high abundant proteins so that smaller ones are, so that it reduces the complexity and it also increases the protein coverage. It's one and the same thing, but written in multiple ways. So, okay, this is about lab based, so we'll skip that. We have not dealt with it. Okay, so that's how, so these were the few questions. Okay, so do you have any questions to ask or anything you want to discuss more? Okay, so try and solve your assignments. If you have any doubts while solving those, you can ask me those questions accordingly in the next week lectures. Okay, so that's it for this week, and thank you for joining. Thank you. Thank you.